So good afternoon. I'm here to update you on what Alex the Leukodystrophy charity is currently doing. So I thought I would give you a short history. So the charity was established in 2004 by a lady called Sarah Hunt. We were then called ALD Life. That was because we supported people affected by ALD and AMN. Sarah decided to set up the charity after her two sons were diagnosed with ALD because there was just no support available for the families. I met Sarah in 2002 when my sons were also diagnosed with ALD, and I joined the Alex TLC team in 2013. Fast forward a few years, and the leukodystrophy specialists in the UK asked if we would consider supporting all leukodystrophies. So after much thought, we decided that it was something we could do. So the charity changed its name in 2019 and became Alex the Leukodystrophy Charity, or Alex TLC for short. We chose the name Alex because both Sarah and I have lost sons who both happen to be called Alex to ALD. Our core strengths at the charity is that we have lived experience. So we understand how it is to actually receive a diagnosis of a leukodystrophy, something you've never heard of, and also to be told that there's no available treatment. So using this experience, we offer individual support. That can be through email, telephone. We also use a lot of Zoom. We also do home visits and advocacy, so we ensure that the families are getting the correct support that they need at the correct time. We have a really good stakeholder network, and as always, patients and carers are absolutely at the centre of everything we do. We work very closely with peer patient groups. Our funders, that's both pharmaceutical companies and others who offer us help. We have a really strong medical professional board, and they help us to disseminate good, reliable information on our website. We work closely with NHS England. We're very lucky in the UK to have the NHS. And we've always striven to try and get involved in research, and I'll talk a bit about that later. We always work collaboratively, so for many years we have worked with organisations such as the ULF and ALD Connect. Another important thing that we do, and a huge part of our work, is to support professionals. So the doctors who are involved in your local care, who may never have heard of a leukodystrophy. So we help them to understand what leukodystrophies are and how it actually feels to live with a condition. We put these doctors in contact with the specialists because, therefore, the specialists can support those doctors in providing your care. Now, for you, those of you that like numbers, we thought to add a few statistics. So by the end of year 2022, we had 1,815 beneficiaries on our database. That may not seem many for those of you in the US, but in the UK, being a much smaller country, that is actually a significant number. We welcomed 112 new beneficiaries, which was a 45% increase year on year. And in fact, 50% of those new families were diagnosed with conditions other than ALD. This proved to us that supporting all leukodystrophies really was important and that we were reaching those families. We engaged in 1,533 support conversations, which was a 73% increase on the year before. Presented about our work at 15 events and held 317 stakeholder meetings. So as you can see, as a very small team, we're extremely busy. We've always been involved in representing the patient voice. This can be within the NHS, with social workers, with pharmaceutical companies, you name it. If we get a chance to actually tell people how it feels to live with these conditions, we will grab it with both hands. We also carefully monitor and evaluate the work that we do. So we use feedback surveys, we talk to our families, because it's really important that we know that we're doing a great job, that's always great, but equally we need to know if we're missing something or if there's something that families would like us to cover. We have a really good group of people who join our steering groups, and they help us when we're thinking of starting projects. We meet with them and we talk through what our thoughts are and ask for their feedback, and also anything that they think should be added. And this really helps to build our relationship with our community. Now, we know that over the years we've supported patients and carers and parents really well, but what about the children? As many of us know, when your child is diagnosed, it's really difficult to have those conversations, to try and explain what your child has been diagnosed with, and to help the siblings to understand as well. It can be a really frightening time for siblings. They often feel cross and angry about the fact that their sibling seems to be getting a lot of attention, and they may not understand why. So we decided to author and publish a book. This book is the first in a series, and it concentrates on adrenal leukodystrophy. We co-developed it with our community. So we spoke to our community, we asked how they had spoken to their children about it. And it's available in digital and hard copy. I've actually got a couple of copies with me, so if anyone would like one, please do come and see me. We also employed 5 new staff members. So 3 to my support team, including me. So that's an engagement and communications officer, Suzanne. She supports me in my work every day and is responsible for engaging with the community, telling them about any events that we're holding, any updates. She just keeps the conversation flowing. Often that's through email, but it's also through, we have a private Facebook group. Now, as we all know, funds. We all need funds to keep our foundations going. So Marlene is our fundraising manager, and she works really hard to build relationships with corporate companies. Her assistant, Fiona, who is also personally affected by leukodystrophy, is just amazing. Another part of my team is Jenna, our counsellor. Again, we're very lucky to have the NHS, and counselling is available, but often the wait can be enormous. It's also very unlikely that you're going to talk to a counsellor who has any idea what a leukodystrophy is. So we took the steps to try and fill that gap, so that when you're talking to a counsellor, she already understands how it feels to live with a leukodystrophy. And last but not least, we received grant funding to employ Christina, who's our research analyst. A really exciting development in the last year is we now have a new NHS England Inherited White Matter Disorder Service and Clinical Patient Registry. So this is a specialised service. We were invited to be the patient representative organisation, and we were an integral part of the development committees. So we really could voice and give suggestions on what this service really needed to look like and what it needed to deliver. We were responsible for developing a survey to determine the patient experience from the registry. So what were their expectations? What were they willing to add to the registry? And these results were used to steer the registry development. One thing that was very clear was that patients really wanted to report their own outcomes, rather than always just relying on clinical data. We also developed patient information for the registry and service, and we will have support staff present at the clinics to give face-to-face support. So the whole idea of this new service is to bring the expertise to the patients, to try and help with the diagnostic odyssey. In the NHS, you can be referred to a specialist, but it used to be quite difficult. But now there's a clear pathway into the new service. And we'll have 5 paediatric centres. There'll be 2 sites in London. Unsurprisingly, one is Great Ormond Street Hospital, and the other is the Evelina Children's Hospital, which is based at Guy's and St Thomas's. The other 3 sites are the children's hospitals in Birmingham, Leeds and Manchester. There'll also be 4 adult centres. London at the National Hospital of Neurology and Neurosurgery will take the lead, and there'll be 3 satellite clinics in Manchester, Leeds and Salford. Now, even although all of the clinics are based in England, it will also allow access for patients in Scotland, Wales and Northern Ireland. So it's really going to pool this expertise and make sure that patients get the correct care, hopefully get a diagnosis and get a diagnosis much more quickly. As I said, the registry will have clinician and patient reported outcomes. So patients and their caregivers will be able to report on treatment outcomes and the challenges and hurdles faced, including the burden of caring for someone. We feel that this is really important because a clinician may, prescribe a medication or a treatment and think that it's really going to work, but you may not see the clinician for another 6 months or a year. So having patients report on how good that has been, or if it's not worked, or if there's side effects, is going to be really important. The registry will include all leukodystrophies. This is going to enable researchers, doctors, to be able to pull condition-specific data, but equally to be able to look for common symptoms and common problems that come across all leukodystrophies. And over time, obviously it's going to take a few years, we will be able to get a good natural history of many leukodystrophies. Again, so important for research and new treatments and therapies. So Alex TLC have always had an excellent reputation for support. We have great stakeholder relationships, we work really well. But we've always wanted to do more in research, but we never had the capacity or the capability. So employing our research analyst has been wonderful. And at the moment, what one of her jobs is sort of looking at the research landscape and making sure that we can disseminate that information to our families in a way that they can understand. In April, we held our Community Weekend Conference. It was our first face-to-face meeting since 2018. It was attended by professionals from all over the world, and we had 180 attendees over the 3 days. Now, talking about research, we were contacted by Professor John Livingston, who is now retired. And he asked us if we would be interested in collaborating with a new group called Leuko Labs. Of course, we said yes. In the UK, we do lots of research, but it does tend to be in individual labs, and it doesn't feel like it's particularly joined up. Leuko Labs' aim is to bring that expertise together. And Dr. Naomi Hamilton is one of the founders, and you're going to hear from her in a minute about a bit more about Leuko Labs. And I just wanted to share a picture of the Alex TLC team.
I'm going to introduce the network, so Leucolabs that Karen talked about, and I will also tell you a little bit about research. The research I do in my lab at the University of York. So this is the dream, having a sick patient and being able to cure that patient. And in terms of genetic diseases, and other diseases too, but here we'll focus on genetic disorders because that's what leukodystrophies are. It's a very, very long and complicated road. So obviously there is a diagnostic and then genetic testing, trying to identify which mutation patient have. And then there is impact on families, and all of those is part of research. And then there is this whirlwind of events that needs to happen before you can have a cure. And this is research on biomarkers, pathology mechanism using many different models, therapy development, and finally, clinical trials in patients. And this is what all researchers want to achieve. That's why we come here at this weekend, at this ULF meeting, is to have impact for our research to reach the patients. So this is my take on the road to impact. Discovery research, you can see, is at the bottom of the dial. This is where most researchers belong. We make discoveries in our labs, doing cutting-edge science with the funding in our team, in our wonderful facilities, and in our academic or industry environment. But in those discoveries, to have an impact, we need to communicate it, we need to connect. We need to reach out all the stakeholders in the leukodystrophy world. And then we need to apply that discovery. So we do trials, we go back to discovery box, we change, we optimize, we build confidence in our product. And then we can finally amplify it, scale it up, and hopefully that can become a clinical trial and a treatment. And in the UK, there's a lot of discoveries, a lot of researchers and clinicians trying to push leukodystrophy research efforts, but like Karen said, it was a little bit disconnected. And this is where Leucolabs comes into play in that second box there, in the Connect box. So this is Leucolabs. So here are the three founders. So there is James in Leeds, Dr. Poulter. He's a geneticist by training, and myself in York, and another colleague, Professor Sweeney in York. And you can see there is a little model attached to our beautiful pictures. So near James, there is a brain in a dish. So he works with organoids. And then by my photo, there is a zebrafish, which I will tell you a little bit more later. And then near Sean's image, there is a fly. So the aim number one of this network was to provide a pipeline of quick models to do what we call functional genomics. So you're being diagnosed with a mutation in a gene, and now we identify so many genes so quickly that those genes are not being studied anywhere in the world. So you're being told that your family member is ill and has a potential leukodystrophy, and this is traumatic enough. But then you're being told that no one is studying this gene, it's ultra rare, and we want to try start solving this. And using quick animals, animals that have a quick, for example, breeding stage, and that we can model those diseases very rapidly in them and generate that data. And the second goal of that network was obviously to connect everybody. So researchers in their laboratory can talk to clinicians, talk to pharmaceutical companies, share ideas, and try to have that communication between the bench and the bed. So you can find all this on our website, but the main goal of Leucolabs is really to connect, to unite everyone and make a plan for leukodystrophy research in the UK for the future. So obviously we're not alone. Karen just introduced Alex TLC, and we work together. And then you also heard about the patient registry, and then the newly commissioned leukodystrophy centers in the UK. So a lot is happening in the UK at the moment. So please watch this space. So now I'm going to tell you a little about my favorite model. And zebrafish are very common. A lot of people have them at home in their aquarium. And if you think zebrafish, you're going to think about this, stripy fish. But what you maybe don't know is that this is the second most used animal model in biomedical research, after the mouse, of course. And we do use the adults occasionally, but what we use is the babies, the embryos and the larvae, because they're transparent. So those babies, those embryos are being pushed out of their mum at one cell stage and left on the pebbles at the bottom of the river, a bit like Nemo, right? And so that means that those fish develop outside of their mum. So you can see every single cell divide, and you can see the brain developing. You can literally take an animal and put it under the microscope and see the brain developing. So this is the transparency during development, and this is what we call ex utero, so outside of the uterus development. And the zebrafish is very easy to modify their genome, modify their genes, their DNA. So we can reproduce mutation that happens in a patient inside the zebrafish very rapidly. And what we can also do, which I will illustrate with movies, hopefully they will work, is that we can label the cells or the tissue that we're interested in different colors, different fluorescent, and we call this reporter lines. So this is an example of a reporter zebrafish. We're zooming into the thymus, which is one of the immune compartment in the body, and you can see immune cells being formed in there, coming in and out of the thymus. So these are labeled with GFP, which is green. And you probably know that there are two important cells in leukodystrophies, and oligodendrocytes that make myelin are very important, and we can label this in zebrafish. So this is a dorsal view of a zebrafish larvae, and this is the myelin that is being formed in the brain by those very bright oligodendrocytes. And this is the spinal cord. I don't know if you can see my arrow on the screen, but so the spinal cord is being myelinated. And we can zoom in, and you can see here the individual very bright oligodendrocytes in the spinal cord of the fish. And you can start seeing those myelin sheath being formed in the spinal cord. And the other cells that are important in the brain are the immune cells, the microglia. And in zebrafish, like I said, we can put them under the microscope and we can just look at those microglia working, doing their job. And here you have a microglia in pink that is clearing up dying cells in the brain. And you can just look at the cells do its normal job. So this is a little bit sped up. It's a lot slower than this. But you can see that cell being attracted by that dying neurons, which is labeled in green, being hoovered up, and then off it goes with the dying cells, and that will be digested, and this is how microglia clear the brain from debris or dying cells, which happens all the time, and I'll come back to this later. So the microglia, you hear about inflammation in the brain, but the microglia really are the guardians of our brain. They're supposed to be there to protect our brain. They have multiple roles. They are sentinel, they can sense danger. They are the housekeepers of our brain. They maintain what we call homeostasis. So any debris that's not supposed to be there will be hoovered up by the microglia. And they're also the warrior, the protector of our brain. If you're being infected by a virus, they will come and they will get rid of it, or they will try. And the first role of the microglia during brain development is this housekeeping, that cleaning job. So I don't know if you know, but during brain development, we make too many neurons. And about 40%, so nearly half of those neurons, during development, I'm talking about a healthy brain development, they will die because they didn't connect properly with the other neurons. They haven't formed what we call the circuit. So they will die. And this, during an organism being developing, calls in those microglia in the brain. So at that point, there is no immune cells in the brain. And they're being called by those dying cells. And this is their first ever mission, their first ever job, coming in the brain and be the cleaner, removing all of those dying neurons. This happens in everyone's brain, in the zebrafish, in the mouse, everyone that has a brain with neurons. We make too many neurons. So we decided to look at this event in normal animal and in leukodystrophy models in zebrafish. So what happens to those microglia? So this is a video of a brain of a zebrafish with healthy microglia, a healthy brain. And you can see they're very active. They go around the brain and they collect those dying neurons like I've shown you before. And then we looked at the brain of our mutants. So this is a mutation for a leukodystrophy gene. And if you look at that video, you can see those big blobs of microglia that are unable to move. And they've eaten so many of those dying cells that it's impossible for them to carry on doing that job. And then what we see over time is accumulation of those dying neurons that they're not being cleared away by those microglia. So this is the hypothesis that we work with in our lab, is maybe there is a common point to all those leukodystrophies. And maybe it's right from the start during brain development. Maybe some of those microglia are defective, dysfunctional at clearing away those dying neurons. So in this particular leukodystrophy mutant that we've developed in the lab, we could prove this, that the microglia are deficient. They really struggle to remove those dying cells, and they start to accumulate, and then they become really engorged, really, really big. So this was a study that was funded by ELA, the European Leukodystrophy Association. And we've used this fish to try to understand the role of the microglia. So these animals, they have white matter lesions in their brain, which we can do with MRI in the brain of the zebrafish. They move a lot less. They have reduced survival. And when we found out that those microglia could potentially be the issue, this is when Holly joined my team, and she wanted to see whether we could replace those deficient microglia. So that's the project of her PhD, which I'm going to tell you in a little nutshell. So in the zebrafish, what she's done is collect healthy microglia or macrophages from a healthy animal, labeled in green. She can purify them, put them into a needle, and inject this into the blood circulation of the sick fish. And then she checks a couple of days later, and you can see there's a lot of green cells in the brain of the fish. And again, because the fish is transparent, it's very easy to see whether your injection has been successful or not. And then she's been looking at whether this injection of a few healthy microglia can help with all the symptoms that the fish had. So the first test was to see whether those microglia can be good cleaners. Can they count them in the brain, and can they get rid of all of those dying neurons? And the answer to this is yes. So this is a normal brain with less dying cells. This is a leukodystrophy brain with a lot of dying cells hanging around. And this is when you add healthy microglia. You can see that number goes down. She could also check that the fish were moving more. So you can see here again, healthy animals move a lot more than the leukodystrophy mutant. And then if you add healthy microglia, they start moving more again. And this is what happens when they're older. So this is a tank of zebrafish. And in there, can you see that little one in the middle, tumbling, spiraling? So this one cannot swim properly anymore. It doesn't, zebrafish are very sociable. They shoal, like we say. They swim together, but this one cannot control its swimming anymore. I'll play it again so you can see. So this is how, when they're little, they move less. But as they grow older, their locomotion becomes worse and worse. And this is this tunneling phenotype that we can easily detect by just looking at the tanks when we go into the aquarium. So now Holly wants to see whether you can rescue this locomotion behavior by changing the microglia. And she wants to look at many other things in a grown-up zebrafish. But when they're grown, they're not transparent anymore. So we need to find other ways to look at the brain. So this spinning image is a cleared brain, so a dissected brain that is being cleared through something I don't really understand. She knows better, just to remove lipids in the brain and make it transparent again. And then you can stain it for whatever cell you want. So here you have macrophages in orange, microglia in purple, and you have the myelin cells in green. And something else that we've been developing is MRI, just like you have when you go to the hospital for the checkup, but MRI on live zebrafish. So MRIs are, as you know, scanners that are very dangerous to mix with water. So we had to come up with a waterproof way to have a live zebrafish intubated with sedative water to stay still in the scanner for an hour and a half and be able to come out of the scanner alive. So a couple of months later, well, we can scan their brain again. And this is something that's going really well. We've managed to have it published. And you can see here, so that whole organ there, this is the brain. So we can acquire beautiful images of our little zebrafish, which is stuck in that beautifully designed live support chamber by my colleague and collaborator. So the tubes are flexible. If you have a slightly larger female, it should accommodate her inside. You have a little sponge to support the tail, a little blanket to keep them moist because they're supposed to be in water. So this is one of the ways that we've developed to be able to follow the efficacy of what we're developing in a fish. So this is my team. There is Holly at the back there, who's finishing her PhD. Lizzie, who's been working with us for a year, sadly is leaving us. Hopefully she'll come back. Come back, Lizzie, if you're listening. And then this is Diogo, who's starting his PhD in October with us. And I'd like to thank all of our funders, and then I'll take questions if I have any. Thank you very much.
Your lab was the first one to be able to take an MRI of a zebrafish, like a live zebrafish. That is so cool.
Yes. So there is another lab that's done it for the heart, but it was a non-recovery procedure. The goal here was to really be able to scan them and then scan them again. So we did a lot of work on, the anesthesia, having something very light. So when they wake up, well, they can wake up.
This is blowing my mind. So instead of air, you're giving them water, like you're intubated with water. And
yes,
then, does it come out through their gills, or what?
Yeah, so it's a very sensitive setup, because you can imagine that any water movement on top of the skull will be picked up, and that ruins the images, as we've experienced many times. Trying to optimize this. So yeah, the game is to have the tubes and the heads as dry as possible. And then the water goes in and then flows out of the gills which are lower down the brain, and then it keeps the rest of the. That's why we had to put them vertical, which they don't like, obviously. It's not a natural position for a fish. But so that's the only way, to have the water flowing out of the gills and keeping them moist for the rest of the experiment.
So I'm absolutely blown away with MRIs of zebrafish, as you know, Naomi. So I'm assuming that the fish are put into a normal MRI machine, so the big MRI machine that humans go into. Oh, can you tell us a bit more about that then?
So patients usually go through a 3T, so this giant MRI machine that you're being wheeled in. But there are two what we call preclinical MRI machines. There is a 7T and a 9.4. And the higher you go in numbers, the more resolution. So because we have a tiny brain, well, me and the fish, we have a tiny brain, we need to have a smaller machine. So it is, yeah, it's smaller and stable. It's still a big machine, but they're not the same. That would be really weird to have a little fish in a giant MRI scanner. Well, thank you very much.
Thank you.
Oh, Marilyn has a question.
It sounded like you were talking about injecting cells, but do you also do like clinical trial stage, before clinical trial stage, where you test out different drugs on the fish?
So this is the next step for this particular model. Now that we've done this shortcut of a bone marrow transplant, because this is what we've done, literally. Yeah. We've just injected the microglia. We didn't inject the stem cells. And now that we're seeing total rescue of most of the symptoms that we see in the fish, now we know which cell we need to target. So developing a new form of transplant, of bone marrow transplant, is not very useful for the community. But if we could find a drug that could help those microglia to function better, I think that would be very powerful. And the advantage of zebrafish at that stage is that they can fit in the same high throughput wells that people use to do drug screening in cells. So those little 96 well plate, you can actually put 3 of those embryos in there. So this is the plan, is to be able to find a drug that could boost activity of those microglia to help them to clear up the brain in those early stages. But first, we need to test whether this therapy still works in all the stages. Is this something transient? It's just helping the first few days and then the disease comes back? Or does it really sustain in the life of the fish?
Okay.
Does that answer your question?
It does. I have a second part, because I always wonder how they find the drugs. Do you do like, is it random, like you have drug libraries, or do you use AI, or do you like just research what the gene function is and then go from that to choose?
So you have a choice. There are so many different drug libraries.
Yeah.
So you can pick kinases, for example, and do a mini screen just on kinases. But I think to be helpful in the situation that we're in terms of the leukodystrophy world, I think FDA approved drugs library will be the first library to go for, something that is potentially being used for another condition that we didn't know had an effect on microglia phagocytic function, so this ability to grab dying cells. I think that would be my first choice, is to go for FDA-approved drug libraries, which we have in the freezer. for in our drug screening facilities.
Okay. Thank you.
When you injected the cells to the fish, were they already showing symptoms, or is this like from birth?
So it's not exactly from birth, but we try to inject the cells when is the time for the macrophages, so the progenitor of the microglia kind of, to enter the brain. So we want to do it so they don't compete.
Okay, so you're preventing the symptoms from developing, but not, like, you haven't tried it on the one fish that was going up and down
And this
to
is,
see if he started swimming straight.
but we kind of know in patient studies that transplanting when the symptoms have started isn't really
Effective,
beneficial.
right?
So we could repeat this in the fish,
But you
but
already
personally,
know.
I don't really believe that it's gonna give us much. I think when the disease has started and the damage is there, this assay is used to identify which cells are driving the early start of the disease. Does that make sense? Thank you for all those questions.